Dear students, we are going to start a new topic and that is entitled as Biochemistry of Extracellular Matrix. Learning objectives of today's lecture, we are going to see biomedical importance or significance of extracellular matrix, and then we would see in detail types, structure and functions of collagen. Extracellular matrix is an extensive network of molecules that surrounds cells, and this is also known as connective tissue. There are three major classes of biomolecules that are found in extracellular matrix. First is structural proteins, which are mostly composed of collagen, elastin, and fibrillin. Then there are some specialized proteins that may contain fibronectin and laminin. And the third important class of biomolecules is proteoglycans, which are also called as GAGs or glycosaminoglycans. So here you can see that how a cell is surrounded by this network of proteins which constitutes extracellular matrix, and these may be collagens, fibronectins, then proteoglycans and laminin proteins. Extracellular matrix has been shown to participate in both normal and abnormal or pathological processes. For example, it is involved in development process. It is also involved in inflammatory processes. It is also responsible for the spread of cancer cells, and this is also involved in the pathology of rheumatoid arthritis and osteoarthritis. Collagen, which is considered as the most important protein of extracellular matrix, if it undergoes genetic disturbances or alterations, then it may lead to the development of two important disorders. One is osteogenesis imperfecta, another is called as Ehlers-Danlos syndrome. Proteoglycans that make up another important category of biomolecules found in extracellular matrix. If get mutated or affected, then they may result into mucopolysaccharidosis, which is a group of genetic disorders. Even during the process of aging, it has been seen that ECM undergoes certain changes. Now, the first protein of extracellular matrix that we are going to deal with is collagen. Collagen is the most abundant protein which is found in the human body. It is a type of a fibrous protein, and this performs many structural functions in the body. It is found in skin, in connective tissue, in the walls of blood vessels, and also in sclera and cornea of the eye. Collagen shows us some special mechanical features that we are going to see later on. Collagen is composed of long and stiff or rigid structure, which is further composed of three polypeptides, which are also called as alpha chains. And these three polypeptide chains are wrapped around one another into a triple helix, which is a rope-like structure. With the help of this figure, you can see that these are three alpha chains of collagens which are interwoven with one another, or these are wrapped around one another to form a rope-like triple helix structure, and therefore this structure becomes very durable and strong. There are more than 25 types of collagen that have been characterized. Even there are some proteins that contain collagen-like domains, and these proteins are referred as non-collagen collagens. Now, the three polypeptide alpha chains that are responsible for making alpha helix of collagen, these are bonded together through hydrogen bonds. And the most common type of collagen is the type 1 that contains two chains of alpha 1 type and one chain of alpha 2 type. This is also written as alpha 1 2 and alpha 2. Then there is type 2 collagen that comprises three alpha 1 chains, and this is also referred as alpha 1 3. According to location of collagen in the body and its associated functions, collagens can be grouped in three categories. First is fibril-forming collagens, second is network-forming collagens, and third is fibril-associated collagens. The very first category of collagens that we are going to study is fibril-forming collagens. These collagens appear like ropes or linear polymers of fibrils. These collagens show special kind of bending pattern when these are observed under electron microscope, and this bending pattern is also called as a regular staggered packing of collagen molecules when these are packed in the form of fibrils. Okay, this figure is showing the bending pattern of staggered arrangement of collagen molecules when seen under the electron microscope. Two types of regions we can see in the collagen molecules. One are those regions which are without gaps. There are no gaps in between them, and these can be stained light, while there are some regions where there are some gaps. So these are the regions which can be stained heavy. So due to heavy and light staining of the collagen molecules, this may appear like dark and light bands. And therefore this structure is also called as staggered arrangement or staggered packing of collagen molecules. Now, the second category of collagens is network forming collagens. These collagens appear like a three-dimensional meshwork. So here you can see a three-dimensional meshwork which forms a network basically. So therefore these are called as network forming collagens. Now the tissue distribution of network forming collagens, type 4 is found in basement membranes, while type 7 is found beneath stratified squamous epithelium. The third category of collagens is fibril-associated collagens. These collagens bind to the surface of collagen fibrils, and as a result of which one fibril can be linked to another fibril, and it can be linked to even other components which are found in the extracellular matrix as well. Now, the tissue distribution of fibril-associated collagens, type 9 is found in cartilaginous tissues, while type 12 is found in tendons, ligaments, and even in some other tissues. Now we are going to start structure of collagen in detail. First, we are going to see its amino acid sequence. Collagen is found enriched with proline and glycine. And both of these amino acids are extremely important for making triple-stranded helix of collagen. Proline helps in the formation of kinks. Now, what are kinks? Kinks are basically twists or curves or bends in the peptide chain of collagens, and hence it is responsible for producing the helical conformation of collagen molecules. So here you can see that these are the kinks, and these kinks are all provided by proline molecules, and this is how the structure can be twisted, or it can attain a shape of helix with the help of proline. Glycine helps promoting hydrogen bonding, and thus it makes the structure of collagen helix stable. And this is also responsible for forming intermolecular crosslinks as well. In collagen, glycine residues are found as a part of important repeating sequence, which is read as Gly-X-Y. So the Gly is for glycine, and this is the fixed amino acid in this sequence. While this X, it can be frequently proline, while this Y, it can be often hydroxyproline, but it can be hydroxylysine as well. So the alpha chains of helix of collagens are called as polytripeptides because these are composed of repeating sequences of three amino acids. And these are also represented as Gly, Pro, and HYP. Gly is for glycine, Pro is for proline, HYP is hydroxyproline, and then triple three. So this is the amino acid sequence of the alpha 1 chain of collagen. So the first amino acid is glycine. Here you can see this glycine is fixed. Now there is X and Y. So here X is leucine, here X is proline, and here is alanine. While Y in this sequence is hydroxyproline, while Y in this repeating sequence is hydroxyproline, while here it is hydroxylysine. So this is how repeated sequence of three amino acids in the collagen structure is repeated, and this is how the structure of different chains of collagens is formed. This is how alpha chains of collagen are bonded together and results into the formation of a triple helix. You can see that some amino acids are exposed at the outer surfaces of chains, and these amino acids are responsible for making bonds with another collagen fibril. Hydroxyproline and hydroxylysine are two non-standard amino acids which are found in collagen mostly, but not found in most other proteins. Through post-translational modifications, proline and lysine amino acids are hydroxylated, and thus these are resulted into hydroxyproline and hydroxylysine. So this figure is showing that how proline can be converted into hydroxyproline. This is the collagen alpha chain, and one of its proline residues has been exposed. Now this proline is going to be hydroxylated with the help of prolyl hydroxylase. This enzyme uses oxygen, alpha-ketoglutarate, ascorbate or vitamin C, and ferrocene. And then it results into the hydroxylation of proline residue along with the production of water, succinate, and carbon dioxide. So this hydroxylated proline residue is then important for making hydrogen bond in the collagen chains, and this is how it can make the structure of collagen stable. Now glycosylation. Glycosylation is observed in the hydroxylysine residues of collagen, and through enzymes, hydroxylysine can be glycosylated. Two important monosaccharides, that is glucose and galactose, can be added to the hydroxylysine residues before the formation of triple helix.